Hi, everyone. Today I'm going to present my project, which is called Precision Nanomedicines for the Treatment of Pediatric Gliomas. So as you all know, the brain is the most complex organ in the body, and it exerts control and regulation over the rest of the organs. The brain is composed by two main types of cells, as you all know, neurons and glial cells. Neurons are those cells able to detect stimuli from the environment and transform these stimuli into electrical signals that can be then sent to other cells as a form of communication. Then on the other hand, we have glial cells, which are cells whose main role is basically to support the neurons. Depending on what these glial cells do, these cells can be classified as astrocytes, oligodendrocytes, or microglia. Now, sometimes these glial cells present abnormalities. They grow more than they should, and as a consequence, they end up forming tumors. Now, when these glial cells form tumors, these tumors are known as gliomas. There are many types of glioma tumors, but my project specifically focused on one that is called diffuse midline glioma. This is a tumor that mainly develops in children and is very aggressive, which means that it grows very fast. And because of that, it's classified as a grade 4 tumor, which is the highest grade. Now, this tumor is found in the brainstem, which is the part that connects the brain with the rest of the central nervous system. This part of the brain is very important because it controls critical functions such as controlling the heart rate or the breathing. And because of that, tumor resection in this area is very difficult. Now, this tumor is characterized by an epigenetic mutation in the histone 3. So as you all know, the histones are these proteins in which the DNA is wrapped so it can fit in the cell nucleus. Because of this epigenetic mutation, there is an alteration in the histone where there is a lysine, there is a mutation. Instead of a lysine at position 27, there is a methionine. And that will alter the interaction between the DNA and the histones, and consequently gene transcription. Nowadays, there is not effective treatment against this tumor. And because of that, sadly, children usually die after 1 year of diagnosis. So this scenario shows that there is an imperative need for the development of new therapeutic strategies. Now, because of the complexity of this tumor, it has been proposed that combination therapy may be very useful to tackle this disease. Now, conventional strategies for screening of drug combinations usually consist on solubilization of drugs in a suitable solvent, and this is then administered to cells when then a response can be evaluated. Now, the problem with this conventional screening approach is that the majority of drugs present poor water solubility, which means that we're going to need to use, in most of the cases, organic solvents to solubilize them, with the consequent and obvious toxicity to cells, and more importantly, to patients. An additional inconvenience of this conventional drug screen approach is that most of these drugs act intracellularly, but just by solubilizing them, they're gonna generally find it difficult to access the intracellular space. So you're gonna, in conclusion, we're gonna see a reduced or a limited action of these drugs. Now, we propose an alternative screening approach where these drugs are not solubilized in any solvent. They're just encapsulated in polymersomes, which are polymeric vesicles. The idea is that by doing that, we will avoid the use of organic solvents, and then we will also provide intracellular drug delivery. Then the drug-loaded polymersomes will be administered to disease cells, and then a response will be evaluated. And we hypothesize that by doing this, we will improve the identification of the real potential of drug combinations. Now, with this in mind, we started preparing our polymeric vesicles or polymersomes. For this, we chose PMPC-PDPA polymersomes. We chose these polymers because they have been used in other cell lines, and they have been shown to be internalized by a variety of cell types by a receptor-mediated endocytosis. Additionally, these polymersomes have shown to encapsulate a variety of drugs, or molecules, such as DNA, proteins, or, for our case, poorly water-soluble drugs. Additionally, and also importantly, these polymersomes are pH sensitive, which are going to help us to deliver the drugs intracellularly. The pH sensitivity comes because of the presence of this amine group in the PDPA block. The pKa of this amine group is found at pH 6.4, which means that for pH lower than that, it will be protonated, and for pH higher than that, it will be deprotonated. The consequence of this is that this polymer, PMPC-PDPA, self-assemble into spherical vesicles at physiological pH. But then when it's internalized by cells, because this process implies a decrease in pH, they will disassemble inside the cells and they will release the drugs. Now, I prepared 3 drug-loaded PMPC-PDPA polymersomes that you see here in this figure. I encapsulated 3 drugs, paclitaxel, carfilzomib, and panovinostat. We chose these drugs because they have been shown to be effective against glioma. Paclitaxel is a vinca alkaloid. It targets tubulin and it stops cells from dividing. Carfilzomib is a proteasome inhibitor, and panovinostat is an epigenetic drug that acts as a histone deacetylase inhibitor. So we characterize this by transmission electron microscopy, among other techniques. And we saw that for the 3 different formulations, we obtained an homogeneous sample of spherical vesicles with a size of around 70 nanometers. Here, the scale bar is 300 nanometers. We also characterize this by HPLC, and we prove that the presence of the 3 different drugs at a concentration of the range of micromolar. Now, after we had characterized our drug-loaded polymersomes, we studied the interaction of these polymersomes with glioma cells. For this, we worked with 3 different glioma cell lines. First, we chose 2 cell lines that resemble the characteristics of diffuse midline glioma, the tumor that we're working with in this project. SF8628 is from human, and line 7 cells are from mice. Then we also work with F98 cells, which are from rat, and they don't present the mutation, but they are from glioblastoma. Now, we first showed that the empty formulations, so empty polymersomes, were safe to use. They were not cytotoxic. And we did that by studying the cell viability after 24 hours of incubation. And we saw that because after treatment with different concentrations of polymersomes, up to 1 milligram per ml, cell viability always remained higher than 75%. Then we also corroborated the presence of the epigenetic mutation in SF8628 and line 7 by Western blot analysis, as expected. And then we also studied the expression of SRB1 receptors, which are the receptors that these polymersomes are going to target in order to gain access via receptor-mediated endocytosis. We did that by Western blot, and we saw that both SF8628 cells and line 7 cells presented higher levels of SRB1 receptors. Now, given the presence of the mutation of interest, together with higher expression of the SRB1 receptor, together with the fact that it was a human cell line, we chose SF8628 for the remainder of our experiments. Now, we wanted to further investigate whether the polymersomes were internalized, so we did that by confocal microscopy. So for those that are not that familiarized with confocal microscopy, in blue, we can see the cell nucleus here. This is the cell membrane, and these are the polymersomes in green. We see them because here they were fluorescently tagged, and this is the merge. So we saw that the polymersomes were internalized as soon as after 5 minutes of incubation, and we also corroborated that by flow cytometry. And what we saw is that the polymersomes were internalized after 30 minutes. Here 30 minutes because was the shortest time point. And then after 24 hours, we saw that around 80% of cells had internalized the polymersomes. Now after this, I started studying the effect of the individual drugs in monotherapy. So what I did is I treated my cells with the different drugs separately, individually. And here on top, we can see the effect of the free drugs, so traditional drug screening. And at the bottom, we see the effect of the individual drugs but encapsulated using the PMPC-PDPA polymersomes. I studied the effect in the form of cell viability. So for those, again, not that familiarized, if we see 100%, that means that that cells are happy, we're not producing any toxicity. And then as we decrease it, we will see white and then red colors. So what we saw is that for similar drug doses or the same drug doses, we were obtaining a higher effect for the encapsulated drugs. And we saw that for paclitaxel and panovinostat. And we see that because for the same doses, we reach lower cell viability values. Then for carfilzomib, we saw a similar effect for both formulations, meaning that for this particular drug, intracellular delivery was not especially an advantage. But we show here that these polymerisms can enhance the efficacy of individual drugs. Now, after that, I started combining the drugs. I had 3 drugs, so in total, I have 3 drug combinations: panovinostat, paclitaxel, panovinostat, carfilzomib, and paclitaxel, carfilzomib. I combined them at different ratios, 5 different ratios, and again, I compared the effect of the free combination versus the effect of the encapsulated combination. And what we saw is that generally the effect of the combinations was higher when the drugs were delivered intracellularly with the PMPC-PDPA polymerisms. Now, this is at 24 hours, but I also measured that at different time points from 30 minutes to 24 hours. Again, these are the 3 different drug combinations. What we saw is that generally the effect, again, was higher when the combinations were delivered with polymerisms, so intracellularly. Not only that, we saw that we see a quicker response. So for instance, if we see panovinostat paclitaxel, we need to wait 24 hours to see an effect with the free combination. But if we use drug-loaded polymerisms, the same combination already show an effect after just 30 minutes of incubation. So we not only improve the effect, we improve the kinetics of response. Now, after that, I wanted to understand whether, okay, we are improving efficacy, but are we also changing the way in which drugs are interacting? So for that, I evaluated the effect of the drug combinations in the form of their synergistic interactions. So here, again, we can see the 3 different combinations. On top, the drugs are free. So this is traditional drug screening. And at the bottom, we see the encapsulated drug combinations. And what we saw is that generally, the synergistic effect for the 3 drugs was higher when we delivered the drugs intracellularly, meaning that we were not only improving their effect, we were also improving their synergistic interactions. And that's it. As a conclusion, we could encapsulate pore water-soluble drugs in PMPC-PDPA polymerisms. These polymerisms improved cytotoxicity of monotherapy and also combination therapy. Also, these PMPC-PDPA polymerisms enhance the synergistic effects of drug combinations. Importantly, these polymerisms allowed us to see synergistic effects that we were not able to see with traditional drug screening strategies. And finally, this methodology can be applied to the panels of drug combinations in order to understand the real synergistic potential of other combined drugs. And that's all from me. Thank you very much for listening. And thank you to Children with Cancer UK for the funding. So now I'll be happy to answer any questions.
Great. Thank you, Belen. It's a really nice talk. And I really like some of the way you're presenting your multi-parameter data. That's really some interesting tools you've got there. Okay, we have a question from Maya about your actually, Maya, do you want to ask a question? You're on.
Yes, thank you, Belen, for this presentation. It was very, very important. I mean, I have a lot of questions. Maybe we can catch up afterwards or in the break with a cup of coffee. Yeah.
Yeah, definitely.
But one of the most important questions, first of all, I'm a fan of combination drugs in one particle. It was that you did. So you put 2 drugs on one polymerism.
No, so
or you combined 2 polymers with different drugs.
Yeah, so we studied both options, either using both drugs, like drug A and B in one single polymerism, or the same formulation. And then an alternative option was to encapsulate first drug A, then drug B, and then put them together. And that's what we did, because
Okay.
we thought it was easier for us to prepare the specific ratios. Because, for instance, if I want to combine drug combinations at different ratios, it's going to be much easier just to have different formulations. Then I can combine them in any way I want.
Great, fantastic. Exactly, that is the answer that I was looking for, but also I was expecting. But also, how can you design, or how do you know that these are the best combinations? I mean, for the translation as well, because now you will start making something eventually that if it goes all the development will reach patients, right?
Yeah.
So how would you know which combination on one particle would be the best, for instance?
But which combination, in the sense of which drugs or which
Ratio.
ratios?
Yeah, no, in the ratio.
So, well, the idea would be that once that is translated, we also maintain this strategy of have them separately, because at the end of the day, the formulations are the same from outside, because the drug is inside. So the pharmacokinetics will be the same in the body.
Okay, so the distribution will be, I mean, they will reach the tumor at the same time.
Yeah, exactly. Yeah.
Okay, that's fine. Thank you very much, and good luck with the rest of the work.
Thank you.
Great. Another question here, Belen, from Che, who's asked, have you identified a kinetic model that suits your system?
A kinetic model?
Yeah. So
no, I haven't looked into that, but it's definitely something worth looking into.
And then a question from Naresh. Have you checked the percentage encapsulation? So are you encapsulating, or are these just polymeric drugs that you're formulating?
I'm encapsulating, yeah. So I can show the figure, or maybe I can just say it, but yeah, what I do is I calculate the amount of, or the average number of drug molecules per polymerisome. So that would be in the order of, let me check. That would be in the order of like from 100 to 10,000 drugs per molecule, and that will vary per formulation.
Great, and just one final question while we're live, and then you've got more questions coming up in the Q&A. And you talked about the HEK27 mutation.
Yeah.
What's the consequence of that? Obviously, it's pretty detrimental to the cell.
Yeah.
But does it drive, does it cause K27 to be in an activated state, so driving transcription, or does it place it in a repressed state, so gene expression is shut off?
Yeah, so what I found is that it's not completely clear yet. So initially, well, there would be, in a normal state, there would be a close interaction between the DNA and the histone, because at that position would be a lysine that is positively charged. And that with the negative charge of the negative charge of the DNA, well, that will cause a close interaction. Now, because that lysine can be methylated, then that positive charge can be suppressed, and that will reduce the interaction between the DNA and the protein. Now, because that lysine is not there anymore, there is a methionine, that transcriptional regulation cannot occur. So that's an alteration that eventually translates into an altered transcription of the DNA.
Absolutely, yeah. It'll be interesting to see if anybody can develop a drug against K27 methionine in the future.
Yeah.
Great, thank you for that. That's a really good talk.
Thanks.